All right, let's jump right into it. Welcome to today's Explainer, where we are quite literally going to unlock the fundamental secrets of life itself. We're going to explore the basics of molecular biology, taking a comprehensive deep dive into how the DNA double helix, RNA, and this incredible process of protein synthesis actually function as the building blocks of, well, everything living. So, whether you're already a biology enthusiast or you're just curious about how life operates down on that microscopic scale, you are definitely in the right place. Seriously, just look at your hands for a second. Every single part of you, your skin, your muscles, even the neurons firing in your brain right at this very moment, is constructed by your cells. But here's the ultimate question, right? How in the world do just 4 tiny chemical molecules hold the exact instructions to build everything about you? To get our heads around this, I want you to think of your cell not just as some microscopic blob, but as a bustling, highly organized, and incredibly efficient microscopic factory. And in this explainer, we're taking a VIP tour of that factory floor. So to navigate the cellular factory, here's our roadmap. We'll cover the basics of molecular biology, unlock the DNA helix, and compare DNA and RNA. Then we'll hit the main assembly line with the central dogma, decipher the genetic code, and finally wrap up with exactly why this matters. Section 1, the basics of molecular biology, studying life on a molecular level. Before we zoom in on the molecules themselves, let's quickly set the stage. What is molecular biology? Essentially, it's the study of biology at its absolute most fundamental level. It's this perfect sweet spot where genetics and biochemistry overlap, helping us figure out how the various systems of a cell interact. Think of the scientists you see here as brilliant detectives trying to solve literally the greatest biological mystery of the 20th century, finding the blueprint of life. In 1944, Oswald Avery demonstrated that genes are actually made of DNA. Then, in 1952, Alfred Hershey and Martha Chase confirmed that the genetic material of viruses is also DNA. And just a year later, in 1953, James Watson and Francis Crick proposed the famous structure of the DNA molecule. In a rapid, thrilling 25-year span, these researchers completely cracked the case. Moving to Section 2, unlocking the DNA helix, the blueprint of life. Now that we know who uncovered the factory's blueprint, let's take a look at its elegant architecture. Every great structure starts with a basic building block, right? Well, for DNA, that block is called a nucleotide. Each nucleotide consists of 3 distinct parts: a phosphate group, a deoxyribose sugar ring, and a nucleobase. And honestly, here's where it gets absolutely mind-blowing. This immensely long chain of DNA that dictates everything about your biology relies on just 4 simple chemical letters, those nucleobases. They are adenine, thymine, guanine, and cytosine. That is it. Just 4 letters code for the entire complexity of life. Okay, so let's look closely at the actual structure these nucleotides form. The DNA double helix has this built-in directionality. Notice the numbering on the sides, 5' and 3'. If you were to sort of walk down one of those sugar-phosphate backbones, you'd encounter a 5' carbon atom, then a 3' carbon, over and over again. But check out the opposite strand. It runs in the exact opposite direction, like traffic on a highway. We call this an anti-parallel structure. Also look right at the center where the bases meet. This perfectly illustrates what's known as Chargaff's rule. Adenine always without fail pairs with thymine, and guanine always pairs with cytosine. They connect via these relatively weak chemical hydrogen bonds, which hold the 2 strands perfectly together. Section 3, comparing DNA and RNA, 2 very essential molecules. So, if DNA is this heavily guarded master blueprint locked away in the manager's office, we clearly need a way to get those instructions out to the factory floor. That is exactly where RNA comes in. The crucial thing here is understanding how RNA actually differs from DNA, because it's those differences that define their distinct jobs. First off, while DNA is highly stable and double-stranded, RNA is generally much less stable and single-stranded. It's really meant to be a temporary messenger. Second, the sugar backbone in DNA is deoxyribose, but in RNA, it's just ribose. And third, and this is a big one to remember, RNA doesn't use the chemical base thymine at all. Instead, it substitutes a different base called uracil. So whenever RNA copies a sequence with an adenine, it naturally pairs it with uracil instead of thymine. Section 4. The central dogma and the flow of information. This is it. This right here is the main assembly line of our cellular factory. It follows a core flow of information in biology, which is aptly named the central dogma. Basically, information flows in one primary direction. It all starts with the genetic material securely stored in your DNA.
When a specific instruction is needed, that section of DNA is copied or transcribed into messenger RNA. Think of this mRNA as a mobile work order. The mRNA then travels out to the cytoplasm where it is translated into proteins. And it's those proteins that actually go out and do the heavy lifting to keep your organism running. So, DNA to RNA to protein.
Let's actually zoom in on that final RNA translation step where the physical product is made, because it is just incredible how fast and complex this microscopic machinery operates. Translation happens inside these cellular machines called ribosomes, and it occurs in 4 incredibly clean, lightning-fast phases. First, you have activation. The correct amino acid is bonded to its specific transport vehicle. Second, initiation. The ribosome locks right onto the messenger RNA to start the whole process. Third is elongation. The ribosome chugs along the RNA, linking amino acids together one by one into a growing chain. And finally, termination. The process stops, and a fully formed protein is released to go do its job.
Section 5. The genetic code, literally the biological language.
Okay, we know the machinery links amino acids based on RNA. But how exactly does it read those work orders? Well, it relies on a nearly universal biological language. It's essentially an exact translation dictionary of life. The translation machinery doesn't read the RNA sequence one letter at a time. It reads it in groups of 3. A 3-nucleotide sequence is called a codon. You can just think of a codon as a single word in the genetic language. Then, small molecules called tRNA carry specific amino acids to the site. They have an anticodon that perfectly matches the mRNA codon. So, for example, if the RNA reads UCU, a specific tRNA drops off the amino acid serine. If it reads GCU, another drops off alanine. This intricate matching game ensures that the protein is built exactly according to that original DNA blueprint.
Section 6. Why this matters. The impact of molecular biology.
We've spent a good amount of time down on the microscopic factory floor today, but let's pull back to the macro level for a second. How does all of this actually impact your everyday life? The absolutely crucial point to take away from all of this is that this system is the literal foundation of your existence. Your genes hold the vital information to build, repair, and maintain every single cell in your body. Furthermore, this factory is unbelievably precise. Cellular proofreading mechanisms ensure that when your DNA replicates, it does so with near-perfect fidelity. And finally, all of this replication, transcription, and protein synthesis, it all exists just to make active agents. Proteins digest your food, they fight off infections, they flex your muscles. This tiny, totally invisible dance of molecules makes you a functioning, living being. Understanding the basics of molecular biology has really brought us to a profound turning point in human history. Because here's the thing. We don't just observe this factory anymore, we are actually learning how to operate the machinery ourselves. We can characterize, isolate, and even manipulate the molecular components of cells now. So I want to leave you with this pretty thought-provoking question. If we now know the alphabet of life, what will we write next? The future implications for medicine, agriculture, and honestly our own evolution are entirely up to us. Thank you so much for joining me on this incredible journey through the cell. If you enjoyed today's explainer and want to keep mastering the basics of molecular biology, please do hit that like button, subscribe to the channel, and visit us online for more deep dive analyses. Until next time, stay curious and keep learning.